Okay, this is the prep for lecture 18, and I was just in the middle of explaining something and I made a big mess of it, so I'm starting over. Um, and what we're going to talk about this time is we looked at sort of complications of single gene conditions, and we looked at, levels of dominance that are not 100%, co-dominance, incomplete dominance. We looked at allelic series, we looked at conditional alleles, and incomplete penetrance, or variable expressivity, and how those complicate looking at single genes. And now we're going to look at, we're going to move to talking about multiple genes. And we're going to touch on single genes as well. So when you make mutations that have a particular phenotype, as you may remember from the previous lecture on allelic series, it is possible to have different alleles of a particular gene that all affect the same phenotype. So you have two phenotypes that are not wild type, and they're affecting eye color in flies or coat color in mice or something like that. And they could both be alleles of the same gene, or they could be different genes that both contribute to that phenotype. Because a trait like coat color, I mean, there's 5 really important genes that contribute to mammalian coat color, and then a bunch of others that modify and refine it, make it, even more variety. And the same with eye color in flies. There's lots, it's just like 80 genes or something that give rise to different eye colors. So when we're looking at a particular phenotype, one of the first things we want to know before we're going to study it is, are these 3 mutations that we're looking at, these 3 different phenotypes, these 3 different strains, are they all affecting the same gene, or are they each affecting a different gene? Because then we're studying just one gene, or we're studying 3 different genes, and then we have to think about how they interact and the different ways they contribute to the phenotype and so on. And so we'll start with complementation. So complementation, does the phenotype we have in this mutant strain, is this from mutating the same gene as from this other strain? Or have we actually identified lots of different genes that influence a particular trait? And then in the second part, we're going to look at sort of biochemical pathways, a little bit about sort of enzymatic pathways, and really focusing on the simplest situation, and then setting the stage for the next lecture, which is all going to be about modified dihybrid ratios. So we'll be working exclusively on 2-gene dihybrid crosses and how different kinds of gene interaction can change those ratios. The genetic ratios are always the same because genes segregate exactly as Mendel said they did, but the way the genes interact can give us some variable ratios, like for example this one, 9:3:4, which is not 9:3:3:1, and we will be talking in lecture 20 a little bit about how that kind of interaction works, and a little bit in lecture 19 too. Okay, so interacting genes and proteins. So I'm going to talk about complementation testing, and I've kind of introduced the topic probably as much as you want at this point, but just the ideas we're asking a question. We've got mutations in a bunch of different strains. They have similar phenotypes. Are they all affecting one gene? So is there really only one gene responsible for eye color in flies, or are there several different genes that all together work to produce that phenotype? And if you're trying to understand a process, the place you begin is how many genes are contributing to that phenotype. So suppose we have a bunch of mutant strains. There are 3 different ones in flies actually, and they all have the exact same phenotype, which the eyes are not the normal dull red, but they're more of a bright, intense orange, almost as bright as that orange flagging tape you sometimes see. They're very beautiful, actually, color. And so suppose you're studying these and you've collected the 3 different strains from the 3 different labs where they were isolated, and you cross 2 of these strains to each other, and you look at the F1 and you see all eyes that are wild type. So you might want to stop at this point and ask yourself why that would be. But we're going to, of course, look at it. When you cross two mutants that have the same or similar phenotypes, they're both affecting the same thing, for example, and you get wild type in the progeny. We call that complementation, and I will explain the term. It's a potentially confusing term because if you don't deeply understand kind of how it works and what it means, superficially it will seem like the name is backwards to what it should be. And so it's good to go through it. I'll be pounding away at it probably more than you want. And then if we cross the two different pure breeding mutants to each other, again with the same or very similar phenotypes, and we find that the progeny have a mutant phenotype, then that's not complementation. It's failure to complement. And we're going to look at the reasons for that right now. Okay, and I'm sorry, I should have erased my ink drawings from the last attempt at recording. Let's just change to blue. Okay, so this is my sort of simple schematic. This is fly. This is the fly head. There's the antennal segment. That's the thorax. I'm not going to draw it again, but I just want to give some orientation. This is the way I just depict different eye colors when I'm talking about eye color. I talk about it in other courses too for different reasons. So we've got two different strains. One we'll call orange 1 and one we'll call orange 2. And you cross them to each other, and the progeny, the F1, are all, they have the same bright orange eye color. And so you should stop at this point, and if this is the genotype of the parents, these here, then what is the genotype of the offspring going to be? And I really recommend, if I say stop and think about this, that you really do that, because it makes you more engaged with the material as opposed to just kind of listening and letting it wash over you, which is very passive, and sometimes not very much ends up sticking. Whereas if you sit and try to figure it out and then get it answered, it's more likely that it will stick in your mind. Okay. Well, that's really great. Okay, so basically this is what we had, and I'm just going to give, this is the genotype you would give it. If the two strains have mutations in the same gene, not necessarily the same mutation, but in the same gene, then they would get one mutant allele from one parent, the other mutant allele from the other parent, and both copies of that gene are still mutant, and that's why you have this this mutant eye phenotype. And I have a diagram that basically shows the same thing. So here's the orange 1 gene at some position on the chromosome. The little asterisk means there's a mutant allele there, a nucleotide difference. In this one, there is a different mutation. These were isolated in different labs, and the gene is probably a couple thousand nucleotides long. It's almost impossible that they would make the exact same mutation, but they could mutate another part of it. And so the asterisk is in a slightly different place and a different color just to indicate that. But when you combine one homolog from each parent, both of the copies of the gene are no good. They're not functioning, they're not producing the enzyme or whatever it is that's needed for wild type eyes. And so since you have two bad copies, we call this failure to complement. And when we see failure to complement, it means that the two mutations have affected the same gene. Now in this case, let's say we take that orange 1 strain just like before, but this time we cross it to the other, the orange 3. That's the 3rd strain. And again, you have wild type now, so you want to figure out what the genotype of the offspring will be. And take, I would suggest taking a couple of minutes. Okay, and so this is the way I'm going to write out the genotypes. When we see complementation, it's a sign that the two mutations are affecting different genes. And so we would write out the genotypes this way to be completely correct. So the orange 1 mutant flies, they have an orange 3 gene too, but theirs is wild type. That's what this little wild type is for. And the same over here. So they're going to be homozygous for a wild type allele of the gene orange 3, but a mutant allele of the gene orange 1. And this is the commonest thing that students have a hard time with, besides just the term complementation, because it's easy to just forget that the other genes are there, because we're only writing down usually the mutant alleles, right? When I wrote it out like this, before we know what the relationship is between those two genes, we just only write the mutant ones. Now that we have this information, we know they're actually affecting different genes, and so we rewrite the genotypes correctly. And the same over here. These guys are mutant for the orange 3 gene, but they're going to have a copy of the orange 1, and it's going to be normal. It's going to be wild type. If it's not mentioned, then it must be wild type, right? We only really mention the mutant ones. So when we cross them, the progeny are going to get one homolog from this parent with the wild type orange 1 and the mutant orange 3, and then one from this parent with the mutant orange 1 and the wild type orange 3, so that there's one good copy of each gene, and therefore enough enzyme is being made for the right pigments to be produced and laid down in the eye to get the wild type. And here's our sort of my picture of it. Mutant for gene 1 but wild type for gene 3, wild type for gene 1 but mutant for gene 3, and then when you combine one of each, you have a good copy of each gene. And that's where the term complementation comes from. You don't want to confuse it with complementation, which is kind of like, oh, you look fantastic today, and you know. Complementation is about having a full complement of something, means you have a complete set of something. And so that's where it comes from. Complementation is referring to completion, essentially. We have a good copy of each gene, so we have a full complement of the proteins that are necessary to make our eye fly eye wild type. And hopefully that's clear. And I think there's just a reminder here. Yeah. When we're doing crosses and stuff, and we write a genotype for an organism, we generally only include the mutant alleles, because there's 13,800 and something genes. We're not going to include all the wild types of the others, but it's just important to remember, and it's something that we're used to doing as geneticists, but you have to learn, of course, when you're first starting out, is to just remember that the wild type alleles of other genes are still present, even though we're not writing them out. Now, I've written them here because, first of all, it's helpful to understand, but also because it's relevant to this discussion, right? So we would include them where they're relevant to a particular cross. And I think, let me just see where we are. I think I will start the next recording with this, because then it takes us into a more complicated thing, which I want to make sure I have enough time to cover properly.